Go.
So in this video, we'll be talking about the anatomy and physiology of the small intestine and the accessory organs associated with the small intestine. So the small intestine has three sections from proximal to distal. They are the duodenum, jejunum, and ileum. And altogether, the small intestine is about 6 meters in length, and it has several different structural modifications that help it increase its surface area because it is probably the most important organ in the alimentary canal as far as absorption and chemical digestion are concerned. Okay, so on this model, we can actually see a part of the small intestine, which is the duodenum. You can see how it's kind of curled around the head of the pancreas. Okay, and I'm going to turn it back so the anterior side is facing you. And you can see one of the structural features that helps increase the surface area of this organ, and that is these circular folds, which are kind of oriented in like a spiraling manner, essentially. So they help kind of guide food through the duodenum and towards the jejunum, okay? And then if we were going to look in more detail at the small intestine, okay, and so if I were going to put this model in this orientation, it would be as if we took out this piece of the wall of the organ and just blew it up so that we could see more of the detail of the wall structure. We can actually see all of the layers of the histological layers of the wall here. So from here to the end is the mucosa. And you can see the surface, the mucosal surface has been altered to have more surface area through the addition of these villi. Okay, and you can't see it on this model, but the individual simple columnar epithelial cells that cover each of these villi also have little extensions of their plasma membrane, which are referred to as microvilli. So the combination of the length of the small intestine, the circular folds, the villi, and the microvilli increase the surface area of the organ about 600-fold compared to what it would be if it was just a long straight tube. It's a pretty impressive set of modifications. Okay, so this particular model is of the duodenum, and we know that because of the structure of the submucosa, which is this section here. And I'm going to turn the model toward you, and you can actually see there's a blue structure here. And this blue structure is representing one of the duodenal glands, okay? And the duodenum needs these glands because it is receiving chyme from the stomach, okay? And so the duodenal glands produce a buffering bicarbonate-rich solution that's going to be added to the chyme as it's coming in through the pyloric sphincter, and this prevents the acidity from damaging the surface of the duodenum, and it also resets the chemical balance of the chyme so that the enzymes that are going to be working in this area can work at peak efficiency. The muscularis externa and the serosa of the small intestine are not really remarkable because they're the same as the base model. Okay? Now, in order for the small intestine to do its job, it needs the support of a couple of accessory organs. And those accessory organs are shown for you here. The two primary ones are the liver, which is shown in brown here, and the pancreas, which is shown in this kind of golden color. And the gallbladder is here. It's also considered an accessory organ, but its functions are really more storage oriented, so it's not a producer of substances the way the other two organs are, okay? So we've just mentioned that we're going to have chyme coming in. Different color here. Chyme coming in from the stomach. And we're going to be, it's still called chyme as it's moving through the small intestine. And we're going to be initially adding those secretions of the duodenal glands to neutralize the acid and get the pH back into the neutral range, okay? So now as the chyme is coming in, let's kind of sum up where we are. We've done some carbohydrate digestion in the oral cavity and the esophagus. We've done some protein digestion in the stomach, and we've done a little bit of lipid digestion in all of those places. Okay, so we're coming in with partially digested carbohydrates, partially digested proteins, and really partially digested lipids. So we've got to finish all of that chemical digestion because this is the main location in the small intestine where the absorption of those nutrients is going to occur, okay, those organic molecules, okay? So we have to first put enzymes in that are going to help us finish that digestion, okay? And that is going to happen through the pancreas, okay? So the pancreas, when we talked about the pancreas in A&P1, we talked about the endocrine functions of the pancreas. Now we're looking at the exocrine pancreas. And the cells that make that up are the acinar cells, or acini, are collections of acinar cells, okay? And these cells produce pretty much every type of enzyme that you would need to digest organic molecules, okay? So they produce a pancreatic amylase. They produce various proteases. In an inactive form. They produce pancreatic lipase. And I'm not going to write it up here, but they also produce enzymes that digest both DNA and RNA. Okay, so literally every type of organic molecule that we have, there is an enzyme produced by the acinar cells that can digest that organic molecule. All right, so we allow those enzymes to become part of a secretion called the pancreatic juice, and the pancreatic juice will be released into the lumen of the duodenum when there are appropriate signals that cause the little sphincter right here, the hepatopancreatic sphincter, to open and allow that pancreatic juice to enter. Okay, so pancreatic amylase, as long as we have the right pH, that should be able to do what it needs to do. Pancreatic, the different proteases, trypsinogen, chymotrypsinogen, etc. They're going to be converted to the active form when they get into the lumen. That helps take care of our protein digestion. But the lipase, we've got the same issue. It has to have something to help it to break up the lipids. Otherwise, it's going to be just as inefficient as the lingual lipase and the gastric lipase. So this is where we have the addition of another substance that is going to be helpful, and that is a substance that's produced by the liver, which is bile. Okay, so bile is a secretion produced by hepatocytes, and we'll look at the hepatocytes in a moment. And they are going to produce bile, and the bile is going to move through a series of ducts that's going to allow it to get closer to the duodenum. Okay, so it passed through the right and left hepatic ducts to enter the common hepatic duct. And if there is no immediate need for the bile, it will be shunted through the cystic duct into the gallbladder for storage of bile, and also to concentrate the bile. If there is a need for bile, the combination of hormones that are released associated with the stimulus for bile secretion will also allow pancreatic juice to enter. And so we'll have a combination of muscular activities in the main pancreatic duct and muscular activity in the gallbladder and this duct system as well. And our hepatopancreatic sphincter will relax, and that will allow the bile to enter. So the bile would be coming from the gallbladder through the cystic duct and then through the bile duct to enter into the lumen of the small intestine, or the duodenum, excuse me. Now, once the bile is present, this is where we're going to get the ability of our lipids to be disrupted. Okay, so if we think about lipids, think about like when you have a bottle of salad dressing, and you're looking at that salad dressing, and the oil is sitting at the top, and the water vinegar mixture with all the spices is usually down at the bottom. Okay, now you don't immediately pour that out, because you know that if you do that, you're going to get nothing but oil on your salad. So you shake that bottle up because you're mixing the lipid in with the water. Okay, that action that you're doing physically is performing an activity called emulsification. And you do that physical activity because you're trying to break up that big clump of lipid and spread it around in that watery material. Now, you can't shake your duodenum that way to get the effect, so you have to use chemicals to do it. And that's where the bile salts in the bile come into play. Bile salts interact with the lipid and help it break up. Okay, so instead of having one huge drop of lipid, that is not going to be efficiently digested, if you add bile, you get lots of lipid droplets. It's the same amount of lipid overall, but just like with the chewing, we've exposed more surface area. Okay? So emulsification, again, you're taking a large layer or drop of lipid and converting it into lots of smaller droplets that mix more effectively with watery fluids, and that gives them more exposure to enzymes. So more efficient chemical digestion, and emulsification is another form of mechanical breakdown. Okay? Now, once the lipid is emulsified, then the digestion by pancreatic lipase will be very efficient, so we'll get good digestion of lipids in the duodenum. Okay? And this converts all of the organic molecules into smaller forms for the lipids. It converts them into substances that can be absorbed for the carbohydrates and proteins, it's converting them into forms that can be acted upon by enzymes in the microvilli of the intestinal cells, and that completes the chemical digestion. The absorption is going to happen through the wall of these epithelial cells, and that allows the end products of digestion. For carbohydrates, that would be monosaccharides. For proteins, that would be amino acids. And for lipids, if we're thinking about triglycerides, that would be monoglycerides and fatty acids. Okay, so those substances are going to move from the lumen through these epithelial cells and into the lamina propria of this mucosa layer. Okay, and depending on which type they are, that will determine whether they go into the blood or the lymph. If it's monosaccharides and amino acids, they'll go into the blood. If it's monoglycerides and fatty acids, they'll move into the lymph of the lacteal and be carried away through that system. Okay, so that summarizes the digestive activities of the small intestine and accessory organs.